First, let's try using the Peak browser tool in ChIP-Atlas. Select ESRRA under TFs as the track type, choose breast as the cell type, and download the bed file. Import the downloaded bed file from the Import button in the Region List panel of Subio Platform. After importing the region list representing ESRRA peak positions, check the details in the regions list at the bottom. The name column contains information about the source, and you can also search by keyword. Extract only the peaks detected in the experiment using MDAMB 231 cells, and save them as a new region list. Load the HG38 genome, and visualize the positions of ESRRA peaks, as well as the peak positions in MDAMB 231 cells. Using Genome Location Filter, you can extract REF-seq genes that have ESRRA peaks detected in MDAMB 231 cells near their TSS. Here, the relative position from the TSS can be set freely. We extracted genes that have peaks in the region 500 BP upstream of the TSS, with an additional 500 BP extension on both sides of that region. After saving the gene list, you can visualize the expression patterns of those genes. When visualized in the genome browser, the expression patterns of genes located relatively close to each other appear to be similar. This suggests that not only ESRRA-mediated transcriptional regulation, but also epigenetic regulation may be involved. Among the genes that have ESRRA peaks near the TSS, filter for genes that pass the QC2 filtering step. When we cluster these genes, the genes with increased expression appear to be relatively common across the three siRNA treatments, whereas many of the genes with decreased expression seem to be specific to SIDE2 or SIDE3. Now, let's look from the opposite direction and use the enrichment analysis tool to examine which TFs have many binding peaks near a gene group showing a specific expression pattern. From the tree, extract the genes that are commonly upregulated across the three siRNA treatments. Copy the gene symbols of these genes. Return to the ChIP-Atlas top page and open the Enrichment Analysis tool. Paste the copied gene symbols into the Enter Dataset section. Leave Dataset B as REF-seq coding genes. Specify the search region as a relative distance from the TSS. After submitting, wait until the calculation is finished, and then download the TSV file. Right-click and choose Save As to save the file as a text file. Open the downloaded text file in Excel. Insert one row at the top, and add a title to the cell type column. Use the filter tool to extract only the experimental data from MDAMB 231 cells. Copy the filtered table, and paste it into another worksheet. Insert one row at the top, and add a title to the gene symbol column. Delete all columns except the gene symbol column, and save the file as a tab delimited file. Import into Subio Platform the gene list of upstream candidate TFs that have many binding peaks near the genes upregulated by siRNA treatment. Cluster these upstream candidate TF genes. Among them, ESRRA was found as a gene whose expression commonly decreases across the 3 siRNA treatments. Return to Excel, press Ctrl+Z several times to restore the original table, and check the enrichment analysis result for ESRRA in the MDAMB 231 cell experiment. It shows that 21 genes are included. Using the Venn diagram tool in Subio Platform, we compared the genes in the upregulated cluster with the genes having ESRRA peaks, and found 14 genes. However, by relaxing the overlap condition in genome location filter from completely to partially, we confirmed that the result becomes 21 genes.